We're going to look at Chapter 20 now, DNA Tools in Biotechnology, in a very abbreviated way, but it does not mean that it's not important. I especially wanted to hit some terminology that I think you'll need in the future. And we talk about biotechnology as the means that we use to make useful products. And this means we're manipulating the organisms, their components, to make things that are useful for humans. Genetic engineering is direct gene manipulation for practical purposes, and we have seen a tremendous amount of that in recent years. This all began once we were able to actually sequence the DNA, which means determine the nucleotide sequence. This was first developed by a fellow named Sanger, but now we have much faster and less expensive methods. When we talk about DNA cloning, that's when you make identical copies of a cell or an organism, so the DNA is identical. Restriction enzymes are used, they're manufactured by bacteria usually, and they cut DNA at very specific sites, and they allow us to split DNA into various links and then analyze what the nucleotides are. And we use gel electrophoresis to separate and visualize these DNA fragments. PCR stands for polymerase chain reaction, and this produces many copies of a specific target sequence of DNA. And so this has been tremendously important in paternity cases and in crime scene cases, because you can take a very small amount of DNA and then amplify it and have plenty to use to identify, is this the criminal that did the particular activity? The key to PCR was finding a very unusual heat-stable DNA polymerase that could be used in the process called Taq polymerase. We now have what is called the CRISPR-Cas9 system, and this is a technique for actually being able to edit and change genes in living cells and organisms. And this is very exciting because we're looking at it as a possible means to repair mutated segments. Down the road, excuse me, the hope is that we might be able to use CRISPR to do things like cure people of things like sickle cell anemia. Stem cells are relatively unspecialized cells, and they can reproduce indefinitely, or under the right conditions, they can differentiate into one or more types of specialized cells. Because they can become multiple types of specialized cells, we call them pluripotent. Totipotent cells, typically seen in plants, they're made from plants, are cells that can generate a completely new organism. And this is really very interesting. This is where a lot of my research was done, where you can take a whole plant, generate from a piece of that plant a cell culture, and then from that cell culture use hormones to regrow a whole plant. It's really very fascinating. Most of you have heard of Dolly, a sheep that was cloned. She died prematurely. Since then, we have cloned many animals, but it's important to remember we're still not sure about the development of these organisms, and we have to remember cloned animals don't always look or behave exactly the same. So these people in Japan and China who are getting hundreds of thousands of dollars to bring back your lost dog that you love so much, that organism is not going to be the same, because not only do we not know what defects might be exhibited, we also know that environment plays a tremendous role in determining what an organism is going to be. Gene therapy is what we call the introduction of genes into the affected individual for therapeutic purposes. We are hoping that we can find more and more uses to treat disorders that are due to one defective gene. And doing something to bone marrow cells these are great candidates because they will be constantly reproducing the new cells that have the correct gene sequence. Biotech is very important because it's also allowed large-scale production of critical products like human insulin and human growth hormone. And, you know, for a lot of people, you don't realize the significance of this anymore, but I remember my aunt, who was a severe diabetic, always used either sheep or pig insulin, usually pig, and periodically, for whatever reason, she would have a reaction to that insulin. Well, now people are receiving bacterially generated human insulin because we were able to put the gene that causes the creation of insulin into bacteria and grow them large scale. Imagine, we don't have to worry about those side effects that are unexpected sometimes because you're actually getting human insulin. Pretty cool. Transgenic is when you introduce genes from one species into the genome of another. It's been in the news a lot. A genetic profile is an individual's unique set of genetic markers, and this allows us to analyze tissue and body fluids, and we can actually analyze these using genetic markers called short tandem repeats. All of us have a unique profile which allows us to be identified by our DNA. Genetic engineering has also modified microbial metabolism, and we're able to use those microbes now to do things like extract minerals like lead from the soil or degrade toxins from the environment. Like if there's an oil spill, we have bacteria now that have been bioengineered to degrade that oil before it can harm the environment. Lots and lots of genetic engineering in plants. We have inserted genes for herbicide and pest resistance. We have inserted genes that allow them to be grown in places where it is more, the salinity is too high for normal growth. Your book doesn't talk about it. We've also bioengineered them to be more drought tolerant. And we've also engineered things like rice and other important food crops to actually increase their nutritional value. Genetically modified organisms, GMOs, we're worried about their safety from the standpoint of allergies and concerns that plants may transfer the inserted genes to wild plants. We feel like that in the future, hopefully, scientists will approach all of this with humility and with great caution to minimize safety concerns.